Today we have two things to do. One will be figure design. We will learn a lot about figure design today. Try to finish today. Let's see how far we can do. And the second thing will be to do experimental. These two things we will try to learn today. This file will be quite big, so I will do this next. Let us start with the experimental part. All of our work, your thesis or your paper, manuscript, should always have this experimental. And experimental is sometimes called materials and methods, okay? So either experimental or materials and method, depending on the journal. Some journals will take this and some journals will take that. But basically they are the same. So you can understand what they are asking. Materials, that means what kind of materials you are using, and methods means what experiments you are using. So basically this is the things you buy and use, and these are the machines or the techniques that you are using. And these are materials means what kind of materials we have. When we write materials, it's not very easy. So let's say we are talking about materials first. What kind of materials you can use? So materials can be chemicals. Can be chemicals that you purchase from different companies. Materials can be softwares. So chemicals, software, or any consumable. In general, we say any consumable that you will use up. So these are the materials. Now when we write materials, we have to write in a certain way. So for example, you are buying, let's say, a chemical called ethanol. So you have to, whatever chemicals you have used in your work, whatever software, whatever consumables, you have to write down. Or it can also be like lab wares. Sometimes you may be using centrifuge tubes. Sometimes you may be using dialysis bags. All of that must be mentioned. So chemical, software, any consumables, labwares should be mentioned. But there is a way to do that. Let's say a very common chemical, ethanol. You have used ethanol to clean something. How to write this? So you say, we purchase ethanol from such and such company. Not only that, so we have to write it like this way. This is the purity of the material. You have to mention this. So ethanol 99.5% was purchased from, let's say, TCI chemicals. So this is the way we write it. So you write the name of the material, the purity, and from which company you have purchased. Now you may be thinking that my work includes a lot of materials, and some of them are bought from this company, some of them from the other company. Okay, let's bring in. So ethanol 99.5%, something else, diethylene glycol, you can tell whatever percentage that is. Then instead of was, you just say where. That's it. So if you buy more materials from the same company, so you say ethanol, comma, DEG, comma, PEG, comma, something, something, were purchased from TCI Chemicals Taiwan. You must know the company name. So remember this. This is the company name, and this is the country. You have to mention this, and this is the purity. So anything that you have used in your work must contain these three information: purity, company, and country. And remember, this company is the original company, not the supplier. So what is the difference between supplier and company? Supplier is, so for example, a supplier, you will see we buy a lot of chemicals from suppliers, and they are in Taiwan. They are called agents. Supplier or agents. Agents. Let's say one of them is called KM3. This is a supplier only, but they can give you materials from across, from Alpha Acer or from the big one, Sigma Aldrich. These are the agents. Don't put the name of the company, this company. And companies are mostly, if it's a chemical company, then it is Sigma. That is the most popular, probably you are using Sigma. And the other one is, so these are the companies that we mostly buy from. Probably your lab also buys from these companies. However, These are the companies, but those are the agents. So you will see this KM3, Uni onwards. These are the agents. Don't put their name, put the company name. So the name that you see on the bottle, you have a bottle and you see that name must come here. So that's the company. Don't use the agent's name. That we purchase this and this from KM3 or Uni onwards or some company, no. And you need the country. So 3 things to remember: purity, company, and country. These 3 things should be there for each chemical that you use. Not only chemical, if you are using something like a petri dish or a centrifuge tube or some kind of glass slide or something, whatever you are using, even like Kim wipes, you take a slide and you dry it, that paper, you need to write. Remember these 3 things, okay? Now you will see, I am talking about materials. Materials may also be like petri dishes, centrifuge tubes, glass slides, and what do you call under the microscope you put? Cover slips, yes. Kim wipes. Whatever you are using and you are buying from a company, you have to know this. Also, wafers, you need to write down in that way. Remember, purity. Of course, for a petri dish, you don't have purity. So remember, so purity, company, country. This information must be there. And the same thing holds for a machine. Now we are going from materials to methods. Let's say your method. Methods deal with machines. And these machines can be small, can be big. Whatever machines. Let's take the very easy example of weighing balance. Our lab always have this kind of weighing balance. But we are not measuring kilogram or like say 500 grams. We are measuring micrograms. So a weighing balance, so you say a weighing balance or a microbalance, again the same formula, this thing, okay? Sagar, our microbalance company name, anybody remember? Oh, give any name, okay? A microbalance company, X5 company from Taiwan was used to weigh the chemicals. So this is each thing that you are using must be written down and written down in this format. So you are thinking, what about the other big machines? Let's go to the big machines. Talk of the big machines that your lab is using. Maybe an OCT machine, maybe an MRI machine, maybe a Raman machine. Okay, different machines have different things. So machines can be divided into 2 groups. One is, and the other one is set up in the lab. One is commercial. The whole machine, you order some company, the company gives you. That's a commercial machine. And the other one is you buy the small parts and then you connect them and make up. So one is a commercial, one is a setup. So let's say we have a commercial machine. How to write that? Now see what I have done. You no need to understand what is this. I just say this is the company name. So when we say just like, oh, I am using iPhone 14 is like that. So that is the iPhone part. This is the model. So this is the model name. And this is the country. So the absorption measurements were performed in a JASCO V770 machine, okay? So this is how you write. If your experiment is OCT, just write down the OCT measurements were performed in a blah, blah, blah. Or if you are working with MRI, you say the MRI measurements or the MRI images were taken in a something, something. Now here is the catch. When you have a machine, you have to know what that machine can do. So for example, you say, I use a clock to take the time. And then you say, okay, I have used a wall clock. The wall clock resolution is 1 second. You cannot say, I measured 15.03. You cannot say, because that requires a much faster machine. So when you say the absorption measurements were performed in this kind of machine, you have to say what range, or you might say what range, okay? Machine in the wavelength range from 190 to 2600 nanometer. So this is the machine, and how much it can measure? It can measure from here to here. Similarly, so you must be thinking that, oh, this is not my machine is something else. Okay, your machine is MRI. So ask yourself, this MRI, what is the most important thing? When you write the MRI machine, we use us, then you have to say, is it a 3 Tesla, 7 Tesla, how much Tesla? For MRI, the most important thing is the magnetic field. So you say, okay, I am using a 3T machine. That is the most important thing. So any of your machines that you are using, you have to tell what is the most important thing. For the absorption measurement, my most important thing is this. For the MRI, the most important thing is the magnetic field. For OCT, the most important thing must be something else. You have to write down that. However, remember, these are commercial machines. I did not do anything. I just ordered, and the company gave me this whole machine. Maybe in most of your lab, you are using a commercial machine. If not, then is a little trouble. So see how we are writing it here. So now I am talking of a setup machine. So a machine that you set up, it's not a commercial machine. You buy a microscope from somewhere, you buy a spectrometer from somewhere, you buy some small part or some electronic card, and then you set it up. So how to write that? So those are called in-house machines. Set up in-house, that is the word. Now remember, this is a machine that I buy part by part. Part A, part B, part C, then I join together. I have to name each part. Uses, for each of the part, the same formula. I name the company. If you have the model name, name the model and the country, okay? So company, model, country. So you will see Olympus company is a microscope. Model name BX43 and country Japan. If you have the other one, company name, model name, country, this is a detector. So for chemicals, you were using purity, company, and country. In machine case, you are using model, a company, model, and country. That is how you have to write it. And because you made it up by yourself, it's not a fully made machine, so I say in-house. Or you can say assembled in-house or assembled. That means you join together, you connect them and make the machine. You need to talk to me. You have to ask me questions if you have any problem. Yeah.
Go back to the consumable part.
Consumable part, okay.
We know that a lot of consumable they have different size. So we have to mention it.
Yes, of course. Aaron is asking that if I am using petri dish can be that big, can be this big, can be very small, or even the glass slide. Even the glass slide, okay? The glass slide can be like this, can be like that, can be like this. So which ones? You have to tell everything. So you might be thinking, oh, it's very difficult to tell everything. Yes, it is difficult, but we need that. Why we need that? Let me tell you why we need that. Why? I am trying, or in my work, I am trying to reproduce some of the experiment that some other people have done. So let's say Wayne, now Wayne is making MOS2 quantum dots, and he is following a paper. So here is the paper, and the paper says we have used 380 degrees, so much time and so much thing, and we have used this much material, that much milligram. So he is trying to follow every bit of that. Only then I can copy or I can mimic an experiment that is done somewhere in America or Japan or somewhere. So for that, I am recreating that experiment in my lab in Taiwan. I need to know every detail. So that much detail you need to put. And why? So normally, we, the professors, we get several manuscripts in a week. Can you please check whether this can be published? So somebody else's manuscript, they come to us, and we see. And then we read the paper, we see, is it good or bad? What are the problems? What are the good things? And then the journal, the author submit the paper to journal, journal send it to us. They ask us one very clear question. They ask us this: are all the experiments reproducible? Reproducible means somebody else can do that experiment. So mostly you know that when you follow the experiment, you do everything the same. Your results are not like them. They put the bottle of the solution and they say, okay, it should look black. Now your experiment is not black, but you have followed everything. So there is some hidden information is there. So why we do this? Write everything in very detail so that the experiments are reproducible. That means an experiment in Taiwan that we do here, the experiment that we do here must be successful somewhere else also. And that requires the details of all the things, even the petri dish size, even the dialysis bags you are using, everything, every small thing. You think, oh, this is any glass slide will do. Yes, any glass slide will do. However, we still need to write. So that is why we do it. So this is in-house. If you are making it up, then you have to name each part of it that you buy from the companies and then join. Remember, this spectrometer or this detector or this microscope, they are not the Raman machine. I joined them to make the Raman machine. So the name of the whole machine and name of the parts are different. If you are doing the same with OCT, you have to do the same thing. So in-house OCT machine using X, Y, Z. And this X name, model, country, company. You have to put it like that. If it is a commercial machine, then you are safe. A MRI machine, MTX30, using 7 Tesla was used. Finish. No problem. It's easier to write about a commercial machine. It takes a few sentences more, 1 or 2 sentences more to write the assembled machine and that kind. So this is how you write about the materials and methods. What about process? Process of making tea. We are trying to make tea. How will I write? It's a process.
Boil the water.
Okay, boil water. So how much water? Some kind of technical writing. You see, I cannot say boil water. So 100 ml of common drinking water, or you can say DI water, or you can say water purchased from Chuanlian. So 100 ml of, I have to name the tea, let's say the most common one. So 30 milligram of Lipton yellow label, you see, amount, that amount of Lipton yellow label USA. So I know Lipton yellow label is only one kind of tea. The solution was kept at 100 degrees for 5 minutes, let's say. The solution was decanted. Decanted means strained. Strained means you have the filter, right? Then all the tea leaves are here, the water falls down. So it's called decanted. So the solution was decanted in a 500 ml clay pot. I have a pot. Because I started with 100 ml, so my pot must be bigger than that. So I take a 500 ml pot and that's my tea. Just hot black tea. And if you want to say that then I add 20 ml of cow milk or some milk powder or how much, you have to mention. So I can prepare this tea anywhere in the world, right? So this is how you write the experiment.
Professor,
Yes.
So we can use the chemistry process.
Past tense, yes, of course. So I said experiments, you should just think like you have done it. So his question is, I am using past tense, boiled, was, I am using past tense. Why I am using past tense? Because when I am writing my manuscript, my experiments are already done. So it was done, and then now I have all the results I am writing. So I follow past tense. Only when I am making a statement, I go present tense. When my T is okay, and then I say the absorption of T is at 500 nanometer. I am making a statement. The absorption of this T I measure using my machine is at 500 nanometer. That's present tense. You can use. Otherwise, mostly past tense is easier to write. In that detail, you have to write. Yes?
So when we use some simulation softwares like console, now how do we write that in the method section? Like, do we write?
I'll come to that. So but it is not yet software. I will tell what we need to write about softwares. So Now, this is my experimental, and we are writing, let's say, 5 or 6 sentences. What if you are following it from a paper? Then how do I write it? Do I need to write that much, or do I need to write in another way? So remember, I am doing the same experiment from a paper. Then how do I write it? First line, the tea was made according to Lee et al. Let's say reference number 5. The first line is I bring in from where I learned to make the tea. So the tea was made according to Lee et al. However, if you finish here, then there is a trouble. What is the trouble? Let's say there is a paper. You have published this paper, and I am reading it. Then I want to know how the tea was made. Then I say, oh, the tea was made according to Lee et al. Where is Lee et al? Then I have to go to the reference, find out, go to the internet, find out. It's very difficult. So what I do is I will write the whole experiment very briefly. So after I write this, the tea was made according to Lee et al., then I will still need to write 1 sentence or 2 sentences. So we start briefly, or in short, or typically. So briefly, in short, or typically, this is how we start. So maybe in that paper, they describe the whole tea making process in a very long way, right? So this much tea, then stir, then keep it for 5 minutes, wait, then take the temperature down to 80 degrees, then stir, do something and mix, very detailed. So we are not writing in that detail. If you want that detail, just go to that paper. But however, I am writing it in a very small way. So briefly, 50 mg of Lipton Black Label was boiled. Boil means 100 degrees, so 100 degree and decanted to obtain a brown, transparent liquid. Just 1 sentence. However, remember, the details are there in that paper. So if you want to say that the MRI experiments was done in this machine or that machine and you are following a paper, first line should be that one, that the OCT, the MRI, the whatever experiment was done according to, and this can be many. If you want to say I have followed 3 papers, just put 5, 6, 7, 5 to 7, no problem. But in that case, don't give any one person's name. So according to previously published literature. You can use any one. If there is only one paper, just one author et al., you can use that. If they are from different people and different papers, then you say previously published literature. So the tea was made according to previously published literature. And then you put some references, 5 to 7 or 10 to 15, whatever you need. However, now this becomes important. So we say briefly something, or in short, or typically, this is done. Now there is another version. What is that version? Yes, I have followed some paper, but in my lab I have done some changes. Then how to write? So you will use the same line. The tea was made according to, with a minor modification. If you want to say major modification, fine, write it. That is a big modification. I follow this, but I change the whole concept of the experiment. That's my contribution to the work. If it is a small change with a minor modification. Now, when you write that, you have to tell what is the modification. Briefly, then again, after this line you start. Briefly, this was taken. However, the time of boiling was limited to 1 minute only. This is the difference I make. Previously, that paper, Lee et al., he was boiling the tea for 5 minutes. But now I say, oh, that's too much. I only need to boil it for 1 minute. That's the difference with a minor modification. What is the modification? That now I just boil it for 1 minute. So this is Lipton Black Label or whatever label. So 50 milligram of Lipton Black Label tea powder was boiled at 100 degrees centigrade and decanted to obtain a brown, transparent liquid. However, remember, this is important. However, comma, the time of boiling was limited to 1 minute only. In that paper, it was 5 minutes, let's say. So that's the modification. So if you are following entirely the paper, you just write the T was made according to previously published literature. If you have done some change, then you write this one, a little change, minor modification, slight modification. Instead of minor, you can write slight. Or if it's a big one, then just write major modification. So this is how we write the experiments. And you write all the experiments one by one. But again, order of the experiments. How will you write? Now, in your thesis or in your manuscript, there are 10 experiments. Which one is number 1, which one is number 2, which one is number 3? So my question is how to write, how to order them. Experiment 1, experiment 2, experiment 3, like that. This will be done exactly as you have done your work. So for example, you are from Professor Kuo, right? Right. So you do OCT a lot, right? But don't mention the OCT machine first. What comes first is your subject. Is it a mouse? Is it a pig? Or is it a fish? That comes first. So first is I take a mouse or pig or whatever. So now I come to a material that is living. You are using some kind of a mouse or some kind of a pig or some kind of cell. How do I write them? They also have a particular way to write. So for example, if you are using mouse, you already know there are 100 kinds of different mouse. So you have to tell them what kind of mouse. So for example, your mouse can be a KM mouse. It's called Kunming mouse. It can be called nude mouse. And there are many groups, like the one we have used is called BALB-C. So there are many kinds of mouse. You have to tell what kind of mouse you have used. I am talking about now in vivo work, some in vivo. Not only this. When you say, okay, I have used BALB-C mouse, then people will ask you how old they are. Very difficult. So this, you say 2 weeks old. Is that enough? No, not enough. What is the weight? 25 plus minus 5 grams. Is it enough? No, it's not enough. Is it a male mouse or a female mouse? You give all information. So before even you start with the machine, so you say, I used BALB-C mouse, 2 weeks old, 25 grams, something, male mouse, and I have 50 mouse, let's say. So that's my subject. Then what do you do with the mouse? You don't take the mouse and put it under the machine, right? You do something. What is that something? So probably you are looking at the ear of the mouse or something. You have to tell what process that mouse went through. Do you make the mouse unconscious? Do you give some medicine to make the mouse sleep? Sometimes, right? You have to tell that. So apply anesthesia, yes. Anesthesia means the chemical, they breathe and then they sleep, right? But that anesthesia, you cannot just say anesthesia. What kind of anesthesia? You have chemicals, that it is a 15% CO2 with some chemical. You have to name that. That has to be there. And then they are sleeping, right? How do you put the mouse under the OCT? You put it like this, like that, like this, standing up, lying down, everything. Remember how you look at the image. That's very important. So you describe that part. Then you say, okay, you are not doing the OCT over the full body. You are just doing this ear, right? So for that, probably you stretch the ear with some clips or do something. And then your microscope comes in, or then your light comes in, right? Everything needs to be done. So that is the subject. Then comes what you do with your OCT. So first you mention all this. Then comes OCT, and then you say, okay, the imaging was done according to blah, 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 et al. And we have used this much range, that much light, that much this. Then that comes in that part. So in the OCT, now you go back here. Now you come here, okay? Now maybe the in-house, you write all this. Not only that, what kind of conditions? So for example, when we write Raman, this is the machine only. How do we do the scanning? We do the scanning, right? So we also write exposure time of 10-second, accumulation of 3 was used. So this is the scanning condition. Your OCT also have a scanning condition. Write the scanning conditions. How you did the scanning, XY scanning, XZ scanning, YZ scanning, what kind of scanning, what kind of time, how did you do, what area. Normally you do, for example, I don't know, is a 100 micron by 100 micron or more, I am not sure. So you have to tell how much area you did the scanning. Everything needs to be there. And remember why? Why is because of that. Somebody in Japan or somewhere need to repeat your experiment. You have to write in that detail. You get the idea, right? But of course, you need to practice how to write it. You need to read 5 or 6 papers in your topic and check how they write. So you need to read, let's say your topic is MRI. Download some papers of MRI, even the papers published from your lab. Then you read how they write. Then you know how to write. Follow those papers and follow this information, then you can write. So when you are writing about Raman, just check our previous paper, how we write them first, how much detail, then you write. Don't try to write on your own at the beginning. You will make a lot of mistakes. So this is the information I give you. You download the paper from other labs or from my lab, then you read, then you write. So reading the previous papers or whatever et al. Papers, then you write, try to write by yourself. Then you will do better. This is your machine and this is your scanning condition. So you have to write. So this is the subject. That means it can be a human being, it can be something. So that's your subject, that's your technique, and that's your scanning condition. You might be thinking, what do I write for my Raman? For your Raman, your subject is R6G, your machine is Raman, and your scan condition is 10 second, 5 accumulation, 532 nanometer laser. Any machine will have this. So if you are doing absorption, it's the same. Absorption of MOS2. So your subject or the material is MOS2. Okay, and doing what? You're doing absorption. And what is your scanning condition?
1,200 nanometers.
In water or in something, that's very important for your machine. Right? In water or in NMP or in ethanol, that's important. So these are the different scanning conditions. Softwares, the same formula. Now you come to software. How will I define a software completely? The same thing. Company, version, country. So Ken, for your case, it will be. However, you may also mention what kind of computer you are using, because some of the computer, the time is very important. So you are using like a workstation or a desktop or a laptop. You can mention that. If you are trying to say, okay, I am just using a common origin software to make my graph. If you are using MATLAB, I don't know what is the version of MATLAB currently. Anybody have any idea? MATLAB, what is the latest version?
2018B.
2018B, okay. You write it like this, and MATLAB is from US, right?
No.
This is how you write softwares. So for software, country, sometimes they also use the year of publication. If it is published in 2020 or 2021, you also write somewhere that this is a 2022 product. This is for softwares. Now, this software is not enough sometimes. This is the basic information. Now, Ken is doing something very difficult. So Ken is using for some second harmonic generation. So when you do the experiment with the software, you have to define the experimental condition, just like MRI or just like OCT. You say in OCT, I take the year of the mouse and then I scan 100 micron by 100 micron. In COMSOL also, they will ask you that what is your model? Then you say, my model is I take a mesh and with my gold nanoparticle inside, you have to mention the size. That is a 1 nanometer mesh, and then I use perfectly matched layer on all sides. You have to read that and see how they write. That's your COMSOL. But how you do the experiment? You are shining light from here. Your electric field is this way. And here is maybe air. Here is air. Here is your gold nanoparticle. Then you make this box and then you run. So everything needs to be mentioned like this. This is just the machine or the software. But then you have to write the scan conditions. This is true for any material, like any experiment. OCT, MRI, ultrasound, PET-CT, whatever you do, you need to do these scan conditions. Scan condition. So your machine, that's your machine. Like, for example, COMSOL now is a machine. And that's your model, how you do it, and your scan condition. Total. Anything will have this. If you are doing OCT, MRI, anything will have these kind of conditions. So when we start our project, today is the 5th week, I think. So 9th week, when we start our project, you open a blank Word file in your computer. So you have a computer, you open Word. The entire file is empty. So we start with experimental. This is easy to write. However, remember, order. Order is very important. So how you did the experiments, the order is very important. And for the order, the first thing that we take is material. So for the order, explain or write about the materials. And the materials include subject. That means if you have mouse or pig or human, even human subjects, they should come here. That's the first. And then comes your techniques. If your techniques are multiple, that means you may have 3 or 4 techniques, write them. That's 2A, 2B, 2C. So for example, in your cases, it can be many, many things. It can be SEM, TEM, XRD, okay, SAED, XPS. This is how you know the materials. Then you apply, maybe you do UV absorption, Raman, then you do fluorescence. So you write accordingly. What I am trying to say, don't write this part ahead of that. You are breaking the order. Unless you have the subject or the material, you cannot do these things. You need to have this, then you do this. That's the order. Don't put it like this. What is first is first. First I take the mouse, then I do the OCT. OCT is not first and then mouse. No. Mouse is first, OCT is next. So you first have your subject, then you do the MRI. Even if it's a human being. So you first take me for a subject. So I am the subject for the MRI, and then here is MRI. So the order is very important. It's a story. Paper writing is nothing but a story. You are writing a story. It has an order. If you break the order, the story is gone. So order them just as you have done the experiment. So in your case, if UV is first, then write it first. If Raman is second, then fluorescence, or if it is like that. But definitely the material comes first. The subject comes first. They come first. Without the subject, you can do nothing. Questions? Yes, Ken.
If I have just software and experiment, which one will be first?
If you just have software and experiment, just as you have done it. You have both, right? As you have done it. So, for example, your material is gold nanoparticle. You try to simulate the plasmon absorption of the gold nanoparticle. So COMSOL comes next. But you are not sure if your simulation data is okay or not. You want to verify. UV comes next. You support that what you got in COMSOL is okay or not. You might say, what is the problem that I take my UNP, then do the UV-Vis. I already know my data. Then I do COMSOL. And I keep doing it, and I keep doing the programming until matches the UVs. That's also okay. There are 2 ways of telling the story. Here is my material, I do this, and here is my theoretically simulated absorption. But I don't know if it is correct. Let me do the experiment. Here is my experiment, and here is my simulation. They match, so it's okay. You can do both ways. This is materials. As I said, name the material, purity, and whatever acronyms you have were purchased from the company country. So these materials were purchased from this. Something else, let's say OA was purchased from Fisher, this one was purchased from Sigma Aldrich, and that group is purchased from here. So this is how you write. So materials and name all the materials, everything, even if you have used DI water. So DI water you get from somewhere, right? So just write DI water was used for all the experiments and not common tap water. So that's not DI. Now you see, this is a process. So as I said, I am following a paper. So such was synthesized or were synthesized, previously published study reference. And then I say with slight modification. What modification? So typically a 3-neck round bottom, and then I take that much and that's this much. So the first line is this. Okay? Previously published, I give a reference. Then I write, I have done some modification, so I will write it later. This is how we do it. So this is a process. And what about experiments? Let me show you some experiments. So materials characterization, the morphology means how the material looks like. was studied by TEM, and the company is Philips, and the model number is this. I forgot this country. Philips is from Holland. I should write Holland. So this is the UV-vis NIR, so that's just V70 in that range. So you see the UCL spectra luminescence. So this is the machine name, company, country, always. I am using a laser which is 980 nanometer laser, where the company, SDL80, and model name and country. So whatever you do, you write like this. And some scan conditions you have to give. So for example, sometimes I am using this power, so I write that power up to 5 watt was used. So whatever details, so that anybody else in the world can do this experiment. We write it in that way. Now we are doing some kind of cytotoxicity. So what kind of cells we are using? So 41 breast cancer cultured in a, what kind of plate? Plates are so many kinds. There is 32, 48, 96. So I used a 96 well plate. So I mentioned that. Then 0.2 ml of this medium containing so many cells were taken. That is the time, that is the temperature, everything in detail we need to mention, okay? So this is a cytotoxicity assay. Most of us, we do this. Everything must be written in great details. So now this is in vitro imaging. So I write in vitro, that means in the cells. So in vitro imaging using so many things. So I tell them what kind of well plate I use, and then what kind of concentration I take. For photothermal imaging, what machine I used, IR thermal camera, this is the model name, the company and country, and what is the laser I used, everything in this detail. So it is very difficult to learn. You just understand here. There is a difference between understand and learning. You understand the process. Then you start to write. Then you will learn how to do it. Just by looking at this, you will only understand. So I am trying to make you understand what level of details. Then you write it, then you will learn. Now we have done CT, not OCT, but just CT. So micro CT system, company, country, model name was utilized to record the CT image. So that would have been enough, but I add more. Standard scanning parameters used were voltage equal to so much, slice thickness so much, field of view so much, gantry rotation that much. This is my information we need to put in for any of your machines that you are using. So whatever be your work is not like me, but your work is different, but I am giving you the idea how details you have to write. If any of you are working with animals, animal handling is also experimental, so you have to know how to write that. This is a typical example of animal handling and tumor induction if you work with this. So the experimental, like this, protocols for animal handling and tumor induction, previously published work, and approved by this iCook. iCook, we call it iCook. So if you want to do work with animals, you need to apply for Institutional Animal Care and Use Committee, which is called iCook of whatever university, and you need to obtain this number before you can work with animals. So that is the first line. And then you see, in this study, 8 female mice, BALP-C, 6 to 8 weeks old, weighing between 20 and 30 grams, were purchased from the whatever National Applied Research Lab, NARL, Taipei, Taiwan. So this is the subject. If your subjects are human, you have to also write like that. So here, in my case, it was mouse. So we wrote how many? So 8 female mice, that much old, that type, weighing this much, where we bought them. Everything is written over here. In your case, if it is human, you have to write in this study 64 or 100, female or male, whatever, that much old, you have to write. And as I was talking, you see how much detail. So how did we keep those mouse? We keep it in the animal center up in the mountain, right? But not only that, so they have to be kept 10 hours in the light and 14 hours in the dark. And what kind of food? So we say standard food was given and provided with DI water on a schedule. How much water? Every little detail we have to write. It's not easy. So we have to read a lot of papers to see what kind of details we need to write. How the animals are kept, how much water we gave, what kind of food. And now we are putting the tumors in the mouse, if you are working with tumors. So subcutaneously means under the skin. Cutin means the skin. So injection under the skin. Subcutaneous injected with 41 cells. That is a breast cancer. How much? 100 microliter of PBS containing 5 times 10 to the power 5 cells on the dorsal side of each mouse, and were left to grow for 2 weeks. So in 2 weeks the tumor will form, and the volume will be between such to such millimeter cube. So this is that detail you have to write. And not only that, so when we do the imaging, for example, so as I was talking, if any of you are using MRI, so you see, we are using a 7 Tesla MRI scanner, PET MR70 Bruker, that's a company, and that's a country. So model, company, country. And how much UCNP we injected, so everything is written over here. And when we do the imaging, that's again, now you see, we have our material, right? So we have to inject. Injection can be like subcutaneous, can be intratumoral. You have the tumor and you inject inside the tumor. And the other one is you have the tail and you inject in the tail vein so that you allow the material to distribute. You are not injecting right inside the tumor. This is normally done when we try to prove targeting. Then we put it through the tail. So you see tail vein injection. So this you need to know the difference. Tail vein injection, intratumoral injection, or subcutaneous injection. 3 kinds of injections are there. You have to know that. So with that detail, and if you are using any kind of software, you have to also write that the images that you get, you know, sometimes the MRI images, you cannot work with PowerPoint. They need particular software. So AMIDE or some other, there are particular softwares to handle. So what kind of software you use for OCT? MATLAB. That's enough. LabVIEW, MATLAB and LabVIEW, okay. So for MRI, they normally use AMIDE. And for other techniques, probably you are using different softwares. We come back to figure designing. As I am telling you from day one, that the most important thing in your paper or thesis are the display items. For what are display items? These are the display items, figures and tables. Whatever kind of figure you have and whatever tables you have. But you have to make these display items to the best as you can. If the figures are good, your writing is bad, people will still read your paper. But if your figures are bad, nobody is going to read your paper. So display items must be of the highest quality. That is required. So I will tell you how to design. And design has a story. This story needs to be told to the readers. There is a story. I mean, for example, look at this. This has A, B, C, D. You plan a figure, okay? A, B, C, D. It's not easy. You have to decide which one to be A, which one to be B, which one is C, and which one is D. So that it makes a story. That is very important. You cannot just say, oh, I have 4 pieces of data, A, B, C, D. No, 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 no. Make the story in order that this is the first. For example, for these 4 of you, you are making nanoparticle, right? So now you have nanoparticle, then you measure their absorption. So you have a choice. So this is absorption. You say this is my absorption, figure A. This is my emission, figure B. And this is something else, the zeta potential, figure C. And then you put figure D, here are the ACM. Now this is not a good design because you have put your material at the end. So it is not telling a story. You need to put the material in A. Only when you have the material, then you can measure absorption, emission, zeta. But you have put all these absorption, emission here and put the material at the end. So this is the opposite story. This is a bad story. So you need to take this here. So when you make A, B, C, D, you need to decide which one is A, which one is B, which one is C, and which one is D to tell the story. That is very important. Now, this is not my subject. This is your subject. That's OCT. I take, for example, and then I put this picture. And we don't know OCT, right? But I might be interested in reading one OCT paper. So I look at this figure. Do you understand this figure? I see something, then I read the caption. It reads, the OCT orthogonal views of freshly excised mouse ear tissue. Freshly excised, that means I just take the tissue right now, fresh. Allowing identification of epidermis, so that E, that is the epidermis, D is the dermis, and something is something. And then I say, well, I really don't understand what is going on. But her challenge will be to make us understand this figure. Your challenge will be to make this figure understandable to them. How we can do that? So that is the challenge that we have. So these are for experts. Who are experts in OCT, they can understand and they can read this and can understand. We cannot. So my point is, we have to design these figures in a way that anybody, scientist, any student of science can understand this. That is our challenge. Let's look at this. Same figure. I see some enhancement. Enhancement means this figure is your data. It is not the enhancement. So what is enhancement? Enhancement are these lines. I put some lines. I put something to tell you more. That is required. On the data, you do something so people understand what you are trying to say. Your writing will be easier. So you can say the red line indicates the epidermis, or the green line tells you the dermis. Then I know, oh, this is the dermis, that is the epidermis. I understand a little bit. I start to understand. So that is what is required. However, I still don't know, is this, it says temporal retina. Retina means in the eye. How do I know how this experiment was done? I cannot visualize the experiment and what this data is about. In that case, if I show you this, now you will see, although we are not from OCT, but we will still understand. You see, that is the mouse sitting over there. And probably here is some kind of anesthesia, something going on. The eyes are open, right? And the mouse is sitting like that, and that's the eye. Now, here is my scanning condition. So I am doing something like this, and then I get some image. Then I have some idea about what this experiment is about. We are not from OCT, but looking at this figure, we have some idea that, oh, this is how it is done. So probably we are scanning like this, and we are getting this kind of data. Only with this figure, it was not sufficient for the non-experts to understand. However, if I do this, probably even the non-experts, and then I put the caption over here, we understand maybe 50% of it. That is the challenge. You make the non-experts understand your figure. It is not required that they will understand 10%, 100%. But even if you can make them understand 50 or 60%, your paper is going to get more citations. If it is easy to read, that's the main point. I show you some examples. Again, vasculature. You do OCT for vasculature. Now, so this is the ear of a mouse. See this picture? Now you see that the vasculature is clearly seen over here. But you must be thinking, so, how they do this experiment. Then I draw a schematic. So as your work, you should do this schematic. So you say, okay, here is my mouse, and here is the ear. I do something. I put some cream and then stretch it and do something, fix the mouse so that it doesn't move. And then I do something over here, you see. Take extra care to prepare the edges where hair is more, you do something. Then you take this image. It's easier to understand. So you see, the machine that she is working on, the machine is only giving her this data. The rest, this one, is probably a microscope or a camera image. This one is a schematic. This is extra work. But with this extra work, we can understand this figure much better. So this extra work is required. This is from a very high impact paper. It's a beautiful figure, you see. It's color coded. The previous one was not color coded. It's a black and white. This one is color coded. And the color code is over here. The color bar is over here. They put it. There are two scale bars. One is the color, and the other one is the size. So that much is 250 microns. So you can understand how much is this part. And they have written very beautiful figure captions. Once you read this, even I can understand what this figure is. However, there are small problems. Maybe it's an experimental because I don't know what is OCT. So if you want to know that, what I would like to say is probably if you don't understand what is going on and I show you this figure, I draw this schematic and I say, okay, this is my experimental setup. And I put the mouse or whatever with the eye over here. Then I scan. Then you mix this figure with that figure. Then you get the whole story. This is how the experiment is done. And there is something over here. So you get this data, do some Fourier transform, and then you get to here. Then you know this, what is going on. You need not know 100%, but at least I understand 50%. That is what is required. So whenever you design your figure, that's the story. This is how I am doing the experiment. That's my data which is being recorded. And in my slice, it looks like this. Extra work is required. So enhancement of data. You see, these writings were not in the data. I have to do it. I have to put some mark. I have to do this. I have to do this. Only then the data becomes a figure. So data is what you get from your machine. And figure is what I present to you is the display item. This is a long path. This is where you come in. So getting the data and just putting it in Origin or MATLAB is not enough. You need to do that to convert your data to figure. Data is uninteresting and for the experts. A figure is for everybody. Everybody scientific. I cannot claim that non-scientific person will understand this one. So figure with enhancement. You have the data from the machine, then you spend time, this is time, and you do enhancement, then it becomes a figure. It has a story. There are many such examples. I will show you how to make those stories. Very difficult to understand what is going on if you just present me this. However, if you present me this, you see how that is designed. I cut the top part. I just show you D and E, but now I am showing you A, B, C, D, and E. Now I understand. That is the ear. That is the vasculature that you see, the blood going through this, how it looks. This is how you are telling a story. And you see how they are telling the story. This is a big thing, ear. So from macroscopic, you are going to microscopic. It's not the other way around. Not from the microscopic to the macroscopic. It is from the macro to the micro. That's the story they are trying to say. So when you have your data, try to present it like this. Then more people will understand what is going on. So it needs a lot of extra work. Enhance your display items. This is your data. OCT image of anterior tooth. So if I show you this, you will say, where is the tooth? I cannot see any tooth because we are inexperienced. We don't know, right? But now if they draw, here is the tooth. You see? That is the enamel. Here is the gum or some part. And now I can see what is going on. So this part is a schematic. That's your data. Data with schematic or schematic with data. They arrange it in a way that you understand which part is which part. But to draw this, maybe you take 2 hours or 3 hours to draw this. That's extra work. I take this image from Reports in Physics, which has an impact of more than 40, I guess. That's a very top-level journal. Then you understand why they can go to 30 impact, 40 impact. Because normally we will just take the data and put it and say, oh, here is the sulcus or there is the enamel. And nobody has any idea the teeth is like this or that or like this. You put this, I understand. So this is one of the enhancement that you can do, explaining your data. I will show you more examples. What do you see here? Something you understand. I mean, don't behave like a non-scientist. You have to behave like a scientist. So there are some materials. And you see these are some kind of liver, MR images of liver. There are some material. And you see, some of them are dark, some of them are very bright, and some of them are like gray. So what do you understand? What I understand, you see, is these are different materials and most of them contain iron and oxygen, iron oxide. And iron oxide is MRI active, I know. So they should give the MR contrast. That means should be bright, very bright. So the brightest image is the best MRI agent. So can you tell me which one is the brightest? You tell me, then I would say rather maybe this or that. So this composition and that is the brightest. So this is my data that I get from the machine. However, you might say, oh, no, not this one. Maybe that one is the brightest. So how do I solve this problem? So I need to quantify the intensity. Whatever is the intensity, I need to quantify. So different material, these are lever giving different MR contrast. And this is pre-injection. Normally they will look dark. And after the injection, they will look bright, bright, bright. And up to a certain time, after that, it will again go down. So this is my data. But however, I am not an MRI person. So I don't understand much. If the authors do this, then I understand. You see, this is the time over here. And that is, they define a signal to noise ratio. So higher the signal, the better it is. Now I clearly know that these are the different materials. You see these ones, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6. Now I am trying to show you that this is not my work. How I look at it. Non-expert looking at MRI data. So I know which one is the highest. The T1 image, the highest signal to noise is probably that one. And that is the red one. What is the red one? Red one is this one. Manganese 1.57, iron 1.43, and oxygen 4. This one. What time? 30 minutes. This one is the brightest. I know from this data. So you see, your data is basically that is the image. From this you get the quantification, the numbers. That number you do a bar diagram. Then I understand everything. So don't put your data only. Do some quantification and place it side by side. Then you understand this figure. And with the caption, I understand everything. You have your data and do something with the data. Remember, these are not data. The data is only this, only this, only this. This writing I need to do in PowerPoint. I put the A, then this becomes a figure. Data to figure, this path. Data is each of these. These are JPG images. Join them together, make them like a line, make the background black. Do these writings, then it becomes a figure. This is a data. The whole thing is a figure. So a lot of work is not easy, but this is the most important. To make your paper or thesis, that's the most important. Data and data quantification, what you would get from here. MRI, who is working with MRI? You understand nothing from this. I only understand this is in vivo imaging and schematic illustration of lung tumor. So these are probably tumors using this kind of material. However, I don't understand that much, that where is the mouse, where is the tumor, where is what. So this is my data, and that's my figure. This you need to either draw by yourself or get it from somewhere so that you don't violate any copyright issues. If you can draw, that's better. Then you inject these nanoparticles from the tail, so that's a tail vein injection. You draw the lungs will probably have a tumor like that, and that's the image. Then I understand. So you see the difference between data and figure. It's not easy. Sometimes you need to draw schematics. So I will tell you that make a schematic here, put the schematic. Which one is A, which one is B, which one is here and which one is there. We need to do that. There are many, many examples. You won't understand some of the data, but if you can do this, then people will understand. Look at this. This is just a photographic image. It helps you so much. Photographic image, and this is a CT. You see how the CT look like, where the broken or the tumor is. The tumor is right over there. That's the enhancement. You see that red circle? If I don't put that and I just say that this is the mouse with the tumor, and I am sure if I see this, I will say, oh, the tumor is somewhere there. That is a mistake we can make. But you see this red circle? That is where the tumor is. This is the figure enhancement. We need to do this. So here is the tumor. Figures need to be made like these. Not only the data. You need to bring in these things. You need to do these arrows. You need to do these things to make it a figure. And it must tell you a story. These are upconversion nanoparticles. They absorb this light. They emit green. They are negatively charged. How to show this as a story? I must start with the image first. So I understand, oh, these are the nanoparticles. And that's my enhancement. I say that the average size of these nanoparticles are like 12 nanometer. My TEM data is only this box. However, the scale bar and this is what I do. So this only shows me these are the nanoparticles. What next? I do more experiments, more high resolution camera. I show each of these is looking like that. So I mark them with a red dash. And they look like hexagons, not circle. From here it looks like circle. But in reality, they are a hexagon. You see how I am making up the story. So normally it looks like this. In detail, it looks like a hexagon. And if you zoom more, you will see the lattice. 0.5 nanometer spacing, it means something for us. Maybe they won't understand. If they look at it, they will know what is going on. So upconversion nanoparticles, I understand. But I don't understand these things. Then I slowly make up my story. Then I show this. This is absorption. Everybody understands what's absorption. And here is wavelength. So I see they absorb 980 nanometer. Here is 980 nanometer. I explain that. Then they emit green. How do I prove it that these nanoparticles give green light? First, I show a picture. Everybody understands. Even they are not from my lab. They understand that here is a bottle containing the nanoparticle. I shine a laser, 980 nanometer, and they will give me green light. So this is a picture. This is the science, that 540 nanometer. So strong emission here, that means green. That green light is over here. I'm making up the story. Zeta potential, they are negatively charged. So I show negatively charged. So that's a zeta potential. Slowly and slowly. And finally, they are citrate modified. So I show by FTIR that they are citrate. This is one figure. This is data. The whole thing is a figure. And in this figure, you will say, oh, come on, what is going on? I see so many things. Put the numbers, and over here I will write something, what these numbers mean. So this is how we design figures. So remember this is a basic structure. I will show you more examples, and we will continue with this. So with that, I think we still have some time, so at least 20 minutes time. I have a writing exercise for you. This is very easy for most of you. Just give them, I will just tell them what to do. This is about the use of conjunctions. Most of you know conjunctions are a word that we use to connect two sentences. So here is one sentence, and here is another sentence. We connect with a word, maybe and, as, for, like that. We connect to two sentences or two meanings of two parts of a sentences. We connect by some word. These are called conjunctions. Mostly we use it, we don't know it, how to use it. So here you will see there are several examples. Conjunctions can be like, for example, and, as, for, although. These are all can be used as conjunctions. Here I give you some sentences, some like 15 sentences. There are some blanks. You put in the conjunctions. That's very simple. It's very easy. Put in the conjunctions that you think is right. These are small things, but is easy to learn. It's basically not English, just your idea. I discussed this word, okay, number 7. Ted didn't feel bad about losing, or he didn't show it anyway. Now, I guess many of you have got this one wrong, right? You have got this was wrong not because of your problem. Actually, whenever we use the word or, we should have in the sentence the word either. So either or, neither nor, they come together. Either or and they come together. If I gave the sentence like this, either Ted didn't feel bad about, he didn't show it. If I gave this sentence like this, I am sure most of you would put or here, because there is a either here. either and or comes together. But just because this was missing, probably many of you made this mistake. So if I said either Ted didn't feel bad about losing or he didn't show it, then the sentence is better. However, you can still imagine that the word either was absent. That's why you probably missed the or. So normally, in a sentence, either and or and neither nor comes together. So if there is an either, in a sentence, if there is an either, there must be an or somewhere in the sentence. So like this, either Ted didn't feel bad about losing or he didn't show it anyway. These two must appear in pair. In this sentence, because the either was absent, so you are confused what to put here. Some of you probably wrote and. If I wrote the either, probably all of you would put or. These are very important. So don't forget the comma there after moreover, therefore, and however. This is technical. Okay, I think that's time enough. Any of you have any questions?